Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University. And our video today is about psoriasis pathogenesis with focusing on the role of keratinocytes in psoriasis pathogenesis. The development of psoriasis is a complex process that arises from the very complicated interactions between keratinocytes, immune cells, and various other cells present in the skin. In recent decades, psoriasis has been increasingly recognized as a disease primarily driven or done by the immune cells, with keratinocytes serving as the key mediators or executors that carry out the immune cell functions within the context of psoriasis. As we all know that the key driver of psoriasis lies within the interleukin-23, interleukin-17 pathogenic axis, which plays a key role in the development and the progression of the disease. Psoriasis is characterized by the abnormal proliferation and differentiation of keratinocytes, along with the infiltration of various inflammatory cells. It was found that keratinocytes contribute to the pathogenesis of psoriasis through the initiation phase and the maintenance phase. So as we see in this diagram, keratinocytes can be stimulated by triggers, and the stressed keratinocytes will release self-nucleotides and antimicrobial peptides to activate plasmacytoid dendritic cells, and here we have the initiation phase of psoriasis. Then this activated plasmacytoid dendritic cells will activate the myeloid dendritic cells with secretions for many cytokines, interleukin-23, tumor necrosis factor alpha, and interleukin-12, and then we have stimulated T helper 17 cells, T helper 22 and T helper 1, with the secretion of many cytokines, interleukin-17 and interleukin-22, and interferon gamma and tumor necrosis factors. These cytokines will activate the keratinocytes, especially interleukin-17, and when these keratinocytes become activated, it will secrete antimicrobial peptides, many cytokines, and other chemokines also to make recruitment of neutrophils and other immune cells. And so we have aggravation of these immune cycles. This, in addition to that, keratinocytes will form epidermal hyperplasia and will share with the endothelial cells and fibroblasts to make tissue reorganization. The success of numerous biologics targeting tumor necrosis factor alpha, interleukin-23, and interleukin-17 in treating psoriasis has been remarkable. However, concerns regarding side effects, safety, loss of efficacy, and relapse following discontinuation of these biologics have pushed researchers to find novel therapeutic approaches. And because recent research findings have indicated that keratinocytes may serve as a triggering and maintaining factors in psoriasis, targeting keratinocytes could be promising in the treatment of psoriasis. So, as an integral part of the innate immune system, keratinocytes have the ability to respond to various triggers. When under stress, keratinocytes release self-nucleotides and antimicrobial peptides, thereby facilitating the activation of plasmacytoid dendritic cells, and this in turn leads to the activation and the maturation of myeloid dendritic cells, which produce key inflammatory cytokines such as interferon alpha and interferon gamma, tumor necrosis factor alpha, and interleukin-1 beta. In addition to their involvement in the initiation phase, keratinocytes also function as amplifier of psoriatic inflammation during the maintenance phase. Upon activation by pro-inflammatory cytokines in a synergetic manner, keratinocytes show heightened proliferation and have the capacity to produce abundant chemokines. These chemokines serve to recruit various leukocytes, including neutrophils, T helper 17 cells, dendritic cells, and macrophages, further contributing to the inflammatory response in psoriasis. Also, keratinocytes are capable of producing antimicrobial peptides such as S100, A7, 8, 9, 10, 12, and human beta defensin 2 and LL37, which play a role in stimulating the innate immunity. Also, keratinocytes release various other inflammatory mediators that contribute to the amplification of inflammation in psoriasis. In conjunction with fibroblasts and endothelial cells, keratinocytes also contribute to tissue reorganization through the activation and proliferation of endothelial cells as well as the deposition of extracellular matrix components. To review again the role of keratinocytes in psoriasis pathogenesis and their share in the initiation and maintenance phase. In the initiation phase, we have here keratinocytes become stressed by triggers, and these stressed keratinocytes will secrete self-nucleotides, antimicrobial peptides, which will activate plasmacytoid dendritic cells, and this in turn will activate myeloid dendritic cells. Then myeloid dendritic cells will secrete interleukin 23, tumor necrosis factor alpha, interleukin 12, and activate T helper 17, T helper 22, and T helper 1 with the secretion of the famous psoriasis cytokines interleukin 17. And then we have also interleukin 22 and tumor necrosis factor alpha, interferon gamma. All these cytokines will activate the keratinocytes, and keratinocytes in turn will share in the maintenance phase by secretion of many chemokines to recruit neutrophils, T helper 17 cells, dendritic cells, macrophages, and also will secrete also cytokines to aggravate the process. Also, keratinocytes will secrete antimicrobial peptides like S100, S7, 8, 9, 12, human beta defensin 2, and LL37. And also these keratinocytes will, under the effect of interleukin 19 and 36, will do epidermal hyperplasia, and also will share with other other cells like endothelial cells and fibroblasts in tissue organization. The interplay between keratinocytes and immune cells, particularly T helper 17 cells, plays a key role in the induction and persistence of psoriasis. This dynamic crosstalk leads to the hyperproliferation and abnormal differentiation of keratinocytes, the expansion of dilated and hyperplastic blood vessels, and the infiltration of various inflammatory cells, including leukocytes. There are various factors that can regulate keratinocytes, including genetic regulations, cytokines and receptors, metabolism, cell signaling, transcription factor, non-coding RNA, antimicrobial peptides, and proteins. Targeting these controlling factors presents promising therapeutic strategies for the treatment of psoriasis. These modulating factors do not act independently, but rather synergetically, exerting their influence through multiple mechanisms to alter the biological behavior of keratinocytes. This interconnected network of interactions links keratinocytes to the development and progression of psoriasis. I will start by the first controlling factors, which are the genetic factors. Epidemiological studies have revealed a strong genetic foundation for psoriasis with a high heritability rate ranging from 60 to 90%. Several candidate causal genes implicated in psoriasis are specifically associated with keratinocytes. In this table, we can see characteristics of some mutations of gene associated with keratinocytes in psoriasis. We have some mutations in the form of gain of function mutations, like what happened with CARD14, which is responsible for activation of NF-kappa B signaling. So when we have gain of function mutations, we'll have more activation of NF-kappa B with more formation of pro-inflammatory cytokines and other mediators. And there are other mutations that include loss of function or reduced function, like what happened with interleukin 36 receptor antagonists. These receptor antagonists, interleukin 36 receptor antagonists, is responsible for inhibition of NF-kappa B signaling. So when we have loss of function of this gene, we will have more activation of NF-kappa B signaling, and so again more production of pro-inflammatory cytokines. Let's talk about the second controlling factors, which are cytokines and receptors. The communication between immune cells and keratinocytes in psoriasis occurs via cytokines and their receptors, playing an important role in the pathogenesis of the disease. Key cytokines such as tumor necrosis factor alpha, interferon gamma, interleukin 23, interleukin 17A, and interleukin 22, predominantly produced by immune cells and activate keratinocytes. This activation sets off a cascade of cell signaling pathways, and which will lead ultimately to excessive proliferation of keratinocytes and the production of antimicrobial proteins, cytokines, chemokines, and growth factors. Tumor necrosis factor alpha, interleukin 17A, and interleukin 23 have significant importance in psoriasis, as therapies specifically targeting these cytokines have demonstrated high efficacy in treating patients. Recently, there has been increasing research focus on cytokines derived already from the keratinocytes or receptors expressed on keratinocytes, indicating their potential role in the pathogenesis of psoriasis. And as we know, interleukin 23, interleukin 17 cytokine axis is recognized as a significant contributor to psoriasis, playing a central role in its pathogenesis. Interleukin 23, primarily produced by immune cells, is thought to be essential for the persistence and proliferation of interleukin 17 producing immune cells. However, it's worth noting that keratinocytes also produce interleukin 23, although the precise role of the keratinocytes derived interleukin 23 in psoriasis remains uncertain. In this diagram, we can see that interleukin 23 is very important for T helper 17 cells to produce interleukin 17, and we can see in the diagram here keratinocytes also produce interleukin 23, but its importance or its function is still unknown. However, we also can see that cytokines derived from or receptors expressed on keratinocytes have great importance in psoriasis. Keratinocytes are critical cytokine responders in psoriasis, as keratinocytes specific deletion of their receptors, such as interleukin 17 receptor A, will prevent psoriasiform lesion in psoriatic mouse model. Interleukin 17 family of cytokines comprises 6 members: interleukin 17A, which is often referred to as interleukin 17, interleukin 17B and C and D and E and F. These cytokines exert their effects through heterodimeric interleukin 17 receptors, among them interleukin 17A, the primary downstream cytokine of interleukin 23, is strongly associated with and extensively studied in the pathogenesis of psoriasis. In summary, interleukin 17A, upon binding to its receptors and keratinocytes, triggers a series of cellular signaling pathways, which in turn leads to the production of antimicrobial peptides such as S100A7, LL37 by keratinocytes, promoting the activation of innate immunity. Also, interleukin 17A stimulates the secretion of chemokines by keratinocytes, facilitating the recruitment of various leukocytes, including neutrophils, T helper 17 cells, myeloid dendritic cells, and macrophages. Also, interleukin 17A induces the expression of multiple pro-inflammatory genes such as interleukin 1 beta, interleukin 6, interleukin 8, and tumor necrosis factor alpha, thereby amplifying the interleukin 23, interleukin 17A axis and establishing feed-forward inflammatory circuits. Also, interleukin 17A can indirectly promote epidermal hyperplasia by induction of the upregulation of interleukin 19 and interleukin 36 in keratinocytes. However, other members of interleukin 17 family have received less attention despite their upregulation in psoriatic lesions. One such member is interleukin 17E or interleukin 25, which is produced by both immune cells and keratinocytes, similar to interleukin 23. The expression of interleukin 17E in the epidermis is increased in psoriasis, primarily due to the upregulation by interleukin 17E. By binding to its receptors, on keratinocytes, interleukin 17E facilitates keratinocyte proliferation and stimulates the production of inflammatory cytokines and chemokines through the activation of STAT3. Interleukin 17C is an additional member of the interleukin 17 family that has been characterized as an epithelial cytokine primarily synthesized by keratinocytes within the skin. Interleukin 17C establishes a self-amplifying loop leading to an augmented synthesis of inflammatory cytokines, chemokines, and antimicrobial peptides by keratinocytes. Consequently, this process facilitates the recruitment of immune cells to the skin. They found that the deletion of interleukin 17 receptor A specifically in keratinocytes led to significant protection against induced psoriasis like dermatitis in mice. Also, on the other hand, the deletion of interleukin 17 receptor A on T cells, neutrophils, or macrophages did not show any notable impact on the development of the disease. Therefore, it can be concluded that keratinocytes serve as the primary target of interleukin 17 in psoriasis. Again, the same diagram we can see here, we have the three types of interleukin 17. Not only the main one which we know, interleukin 17A, but also we have interleukin 17E and interleukin 17C, and these cytokines can be produced by keratinocytes as well as interleukin 23, as I previously said. And as I said, that deletion of the receptor for interleukin 17A prevent the effect or the having psoriasis-like dermatitis in mice, while deletion of these receptors on other immune cells didn't prevent the development of psoriasis-like dermatitis. Now I'm going to talk about another important cytokine in psoriasis, which is interleukin 36. Interleukin 36 family, which belongs to the interleukin-1 superfamily, consists of three agonists, which are interleukin 36 alpha and beta and gamma, and one antagonist, interleukin 36 receptor antagonist. All members of this family bind to heterodimeric receptor complexes composed of interleukin 36 receptor and interleukin-1 receptor accessory protein. Currently, the cytokines belonging to the interleukin 36 family are being recognized as the key contributors to the development of psoriasis. The absence of interleukin 36 receptor antagonists resulted in an exacerbation of psoriasiform lesions and also in mice, and also mice lacking interleukin 36 alpha but not interleukin 36 beta or gamma show a reduction in the psoriasis-like phenotype. When interleukin 36 receptor was specifically deleted in keratinocytes, a similar level of protection from psoriasis was observed as seen in global interleukin 36 receptor deficiency. This finding suggests that keratinocytes are the primary cell type responsive to interleukin 36 signaling in the context of psoriasis. Interleukin 36 plays a pathogenic role by stimulating keratinocyte proliferation and intensifying the production of inflammatory cytokines and chemokines, thus amplifying the inflammatory response in psoriasis. Recently, therapeutic approaches targeting interleukin 36 have been developed for the treatment of psoriasis and are currently undergoing clinical trials. Interleukin 22 is a significant cytokine downstream of interleukin 23 and is primarily synthesized by CD4 positive T cells and group 3 innate lymphoid cells. The receptor for interleukin 22, known as interleukin 22 receptor, is expressed on non-hematopoietic cells such as keratinocytes, epithelial cells, and hepatocytes. Interleukin 22 exerts its pathogenic effect in psoriasis by inhibiting the terminal differentiation of keratinocytes and simultaneously inducing the production of antimicrobial peptides and pro-inflammatory chemokines. Interleukin 22 binding protein acts as a natural inhibitor of interleukin 22 by specifically binding to it, thereby suppressing its biological activity. Deficiency in the interleukin 22 binding protein either genetically or through the use of anti-interleukin 22 binding protein neutralizing antibodies, resulted in exacerbation of induced psoriasis-like skin disease. This exacerbation was characterized by increased epidermal thickening and elevated expression of inflammatory cytokines and interleukin 22 inducible antimicrobial peptides. In addition to tumor necrosis factor alpha, another notable cytokine within the tumor necrosis factor superfamily known as tumor necrosis factor like weak inducer of apoptosis, or TWEAK, has emerged as a significant player in psoriasis. Recent research conducted demonstrated that the absence of this tumor necrosis factor like weak inducer of apoptosis led to the alleviation of induced psoriasis dermatitis. Also, mice lacking fibroblast growth factor inducible 14, the receptor for tumor necrosis factor like weak inducer of apoptosis, also resulted in a reduction in disease severity. By going again to the same diagram, we can see clearly how is the key roles played by keratinocytes in the pathogenesis of psoriasis, where keratinocytes can produce many cytokines, chemokines, and antimicrobial peptides. And from these cytokines, keratinocytes secrete interleukin 23, interleukin 17E and C. And also keratinocytes contain many receptors that are vital in the responses to different cytokines by the immune cells. So we can see here we have different receptors for interleukin 17A and interleukin 17E and interleukin 17C. We have here interleukin 36 with its receptor, and we have here what's called tumor necrosis factor like weak inducer of apoptosis and its receptor. And we have also interleukin 22 and interleukin and its receptors and the binding protein, interleukin 22 binding protein that prevent attachment of interleukin 22 to its receptors. So here is the main kitchen for the process of executing the orders coming from the immune cells in the process of psoriasis pathogenesis. So this will keratinocytes not only helping in initiation but also in the maintenance and in the continuation of the pathogenesis of psoriasis. Now I'm going to talk about another regulator factor, which is metabolic. A prominent feature of psoriasis is the excessive proliferation of keratinocytes, which demands a significant amount of energy, amino acids, nucleotides, and lipids. Recent studies have highlighted the importance of metabolism, particularly in keratinocytes, in the pathogenesis of psoriasis. Among various metabolic pathways, glucose metabolism is an important mechanism in psoriasis development. Glucose serves as the primary source of energy for all cells, particularly for rapidly dividing cells, and its involvement in psoriasis has attract more attention. As regards cell signaling, the signaling pathway is a key regulator of psoriasis, affecting various biological processes impacting both immune cells and keratinocytes. Several key signaling pathways are dysregulated in psoriasis, including the signal transducer and activator of transcription, STAT pathway, nuclear factor kappa B, and mutagen activated protein kinase, or MAPK pathways. Among these also, the JAK Janus kinase STAT signaling pathway has been identified as playing a critical role in psoriasis pathogenesis. In keratinocytes, STAT3 has a central role in response to various inflammatory cytokines such as interleukin 6, 17, 21, interleukin 19, 22, etc. STAT3 activation in keratinocytes inhibit cell differentiation and promote proliferation and production of antimicrobial peptides. Also, increasing evidence has shown that NF-kappa B signaling contributes to the pathogens of psoriasis by acting on immune cells and keratinocytes. Actually, the researchers found that NF-kappa B was highly activated in lesional skin of psoriatic patients. Mutagen-activated protein kinases are involved in the pathogens of psoriasis and play important roles in regulating keratinocyte proliferation and immune responses. As regard JAK-STAT pathway and details about Janus kinase and Janus kinase inhibitors, I already have discussed it in other videos, so you can return back to these videos if you want detailed information. And in addition to the transcription factors associated with major signaling pathways that I have discussed, there are other transcription factors expressed in keratinocytes that have emerged as significant regulators in psoriasis. These transcription factors hold promising therapeutic potential in the treatment of psoriasis. Nuclear factor erythroid 2 related factor 2 or NRF2 emerges as a critical regulator of cellular defense against stressors in psoriasis, with heightened expression and activation observed in psoriatic epidermis. The upregulation of NRF2 contributes to keratinocytes proliferation by promoting the expression of hyperproliferation associated keratins such as keratin 6, 16, and 17. Additionally, the AP1 transcription factor superfamily plays a vital role in psoriasis pathogenesis. As regards non-coding RNAs, in addition to genetic factors, epigenetic regulation plays a significant role in psoriasis. Non-coding RNA regulation is an important aspect of epigenetic regulation that affects various biological processes and disease pathogenesis. The role of non-coding RNAs, particularly microRNAs in psoriasis, has been extensively studied in many researches. MicroRNAs are short non-coding RNAs, and they are key players in the regulatory network of psoriasis as they exert post-translational control over protein coding gene expression. Their dysregulation is closely associated with the development of psoriasis. Extensive research has identified over 250 microRNAs with apparent expression patterns in lesional skin of psoriatic patients, which highlight their significance in modulating the functions of various cell types, including keratinocytes and leukocytes, as well as their strong interplay. MicroRNAs play key roles in essential aspects of psoriasis, such as regulating keratinocytes proliferation, differentiation, apoptosis, and inflammation. Long non-coding RNAs are a group of non-protein coding transcripts that are more than 200 nucleotides in length and regulate gene expression at post-transcriptional and post-transcriptional levels. Long non-coding RNAs influence the proliferation, apoptosis, and inflammatory responses of keratinocytes in psoriasis. Identifying dysregulated long non-coding RNAs and the related networks involved in psoriasis will be an important area of research. If you need to know more detailed information about microRNA and long non-coding RNAs, you can return back to my video about epigenetics. Antimicrobial peptides such as LL37, beta defensins, and S100 proteins are small proteins known for their role in activating the innate immune response and their involvement in the pathogenesis of psoriasis. Recent studies have demonstrated that these antimicrobial peptides can also modulate the function of keratinocytes and contribute to the pathogenesis of psoriasis. Also, recent findings highlight the critical involvement of protein dysregulation, such as upregulation of the ubiquitin proteasome system and downregulation of galactin 3 in the development of psoriasis. In this video, I tried to summarize most of the information that came in this nice paper in Cell Death and Disease that was published in January 2022, and its title is Advances in the Pathogenesis of Psoriasis from Keratinocyte Perspective. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.